Welcome student for today's lecture. Student, today we are going to see the next point of our topic, that is the genetic code. Actually आपण प्रोटीन सिंथेसिस पाहत होतो, बरोबर आहे. आणि प्रोटीन सिंथेसिस मध्ये आपण पार्ट फर्स्ट, पार्ट सेकंड आणि पार्ट थर्ड पर्यंत आपण ट्रान्सक्रिप्शन प्रोसेस पाहिली आहे. आता प्रोटीन सिंथेसिसचा राहिलेला पार्ट हा पुढे आपण अजून ट्रान्सलेशन प्रोसेस बघणार आहोत. पण त्याच्या अगोदर जेनेटिक कोड हा कन्सेप्ट तुम्हाला समजणं गरजेचं आहे. त्यामुळे इथं आपल्या सिलेबसमध्ये जेनेटिक कोड हे दिलेले आहे. ठीक आहे. तर आता आपण सुरुवातीला कॅरेक्टरिस्टिक्स ऑफ द जेनेटिक कोड or the properties of the genetic code. So what are the characteristics of the genetic code? So genetic code means what that is. The genetic code is are mainly in the form of the triplet and they are the always present in the mRNA. म्हणजे हे genetic codes जे आहेत, these genetic code where they are present, they are present on mRNA. They are present in the mRNA. So if we read, that is the genetic code, these are read that is from the 5 prime end to the 3 prime end. So it is the genetic code which are present.
Piece of genetic code. 1. The code is triplet. 61 codons code for amino acids and 3 codons do not code for any amino acids. Hence, they function as top codons. Example: UUU, phenylalanine, GGG, glycine. 2. The code is degenerate. In triplet code, 64 codons are possible. Thus, each amino acid is coded by more than one codon. Example: serine is coded by UCU, UCC, UCA, UCG, AGU, and AGC. 3. The code is non-overlapping. The code should be always read in groups of 3 nucleotides from a fixed starting point. The 3 letters of the codon are considered as a unit and should be read as if they are exposed through a reading frame. 4. The code is commaless. In between triplet codes, no letters are wasted. No punctuations are needed between any 2 codons. 5. The code is non-ambiguous. One codon codes for only one amino acids. Hence, it is specific and non-ambiguous. 6. The code is universal. The same code applies in all kinds of living organisms. Example: UUU codes for phenylalanine in all living organisms. 7. Initiation codons. The codons which initiate the process of polypeptide chain formation are referred as starting or initiation codons. AUG is a starting codon and is present as triplet codon on all mRNA. AUG codes for methionine, MET. 8. Termination codons. The triplet code which is at the end of mRNA and does not specify for any amino acid and serves for termination of polypeptide synthesis is the termination codon. The codons UAA, UAG, and UGA are termination codons. UAA is called ochre, UAG amber, and UGA as opal.
In RNA, that is thymine is absent. Okay, so see that is here. The mRNA, if we see RNA, that is in RNA there are the 3 types: mRNA, rRNA, and tRNA. Okay, so here in the mRNA, that is here there are the 4 nucleotides, that is the adenine nucleotide, guanine nucleotide, cytosine, and uracil. That is thymine is absent. Thymine is absent in the RNA. So here if we read, that is in the DNA, if we see in the DNA, that is in the DNA purines, these are the adenine and guanine, and pyrimids, these are the thymine and cytosine. Okay, so here in the DNA, that is genetic code, these are read, that is based on these 4 nucleotides, adenine, guanine, thymine and cytosine. While in RNA, that is adenine, guanine, cytosine and uracil is there. Uracil is there. That is here in the DNA, thymine is present. In RNA, thymine is replaced by the uracil. Okay, students. So see, that is the characteristics of the genetic code. So genetic code, one by one आपण एक characteristics बघूया. So first characteristic, that is the genetic code are in triplet. Triplet, that is triplet means what? In every living organism, that is genetic codes are always triplet. So if we see, that is the mRNA. So see here the mRNA. Okay. So in mRNA, here the first 5 prime end, here is the 3 prime end. So genetic code, if we see, that is a ring, that is from the 5 prime end to the 3 prime end, and the 3 successive nitrogen bases which form the one codon. 3 successive nitrogen bases, that is, for example, at the 5 prime end, here is the nitrogen base, that is the AUG. So here AUG, these are the 3 successive nitrogen base, and these 3 successive nitrogen base which produce the one codon. That is here these 3 successive nitrogen base. It is known as the codon. And let's come into upon codon. So here the codon, these are always triplet form. That is 3 successive nucleotides. म्हणजे 3 successive nucleotide मिळून एक codon तयार होतो. So the codon, these are, that is the first characteristic, that is the codons are in triplet form. That is the codons, that is genetic code is always triplet. पहिला characteristic काय आहे, genetic code is always triplet. And this genetic code, these are read, that is from the 5 prime end to the 3 prime end. Okay. Then next see, that is here. आता आपण हे जर बघितलं तर हा एक जेनेटिक कोड झाला, याच्या पुढे दुसरा येईल, फॉर एक्झाम्पल CGU, हा दुसरा असेल जेनेटिक कोड. It is also in the triplet form. Okay, so each codon, this codon is in the triplet form. This codon is the triplet form. That is 3 successive nitrogen bases which forms the codon. So the genetic code is always triplet. Then next that is here. Second characteristic, that is about the polarity. That is genetic code having polarity. Second characteristic, genetic code having polarity. Genetic code having polarity. मग genetic code मध्ये polarity काय असते? If we see, that is the genetic code, these are always in direction, that is from 5 prime end to the 3 prime end. म्हणजे हा 5 prime end झाला mRNA चा, it is the 3 prime end. Okay, so these are read from the 5 prime end to the 3 prime end. Otherwise the message will be changed. जर समजा एखादा जर genetic code change झाला, म्हणजे for example AUG, it is the initiation codon. AUG. Okay, it is the initiation codon. So AUG, it codes for amino acid. AUG codes for amino acid. कोणता amino acid कोड करतं? Methionine. Codes the amino acid methionine. If there will be the change, that is, if this AUG is changed GUA, या पद्धतीने जर ते चेंज झालं, or if we read the code, that is the GUA, okay, So polarity means what? That is, here the first codon, second codon and third codon, and these codons are always from 5' end to the 3' end. So here they having the polarity, that is the genetic code always in the direction, that is here 5' end to the 3' end. जर आपण 3' end to the 5' end रीड करत गेलो तर हाच AUG हा कोणता अमायनो ऍसिड कोड करतो? मिथिओन आहे. जर आपण हा असा रीड केला GUA, that is it will be the wrong. It will be the wrong. Okay, so genetic code having polarity, that is they are read from 5' end to the 3' end. Then next characteristic, that is genetic code is non-overlapping. बघा आपण पहिलं ट्रिपलेट बघितला, दुसरा पोलॅरिटी बघितला, तिसरा आहे that is genetic code are non-overlapping. Non-overlapping. Genetic code are non-overlapping. So see, that is here if we see, that is experimentally evidence who proved that, that is the genetic codon, these are the non-overlapping. So see, that is how they are non-overlapping. So see here, that is here is the 5' end, here is the 3' end, that is of the mRNA. So here the first genetic code, that is, for example, here the first genetic code is the ACG. Okay, and second genetic code will be the here CGU. Okay, so here ACG, this genetic code, it mainly specify the single amino acid. Single codon specify single amino acid. Okay, so see, that is here the ACG, it code, that is the amino acid AA1, while CGU, this codon codes amino acid AA2. So it is the one amino acid, it is the second amino acid. Like this, one codon specify one amino acid. So here the experimentally evidence is, that is for the overlapping codons. So here on the mRNA, if there are the 6 codons are present, first 3 codon, that is they code one amino acid, another 3 they code the another amino acid, like this. Okay, so these are the codons which are non-overlapping. Now overlapping condition मध्ये कसं असेल बघा? In the overlapping condition, that is here it is the 5' end, here is the 3' end. Okay, now overlapping condition मध्ये कसं असेल? So see, that is here ACG. Okay, so here is the CG and U. CG and U. So here in the first codon, here like this. So it is the first codon which will be called the one amino acid. So overlapping म्हणजे कसं येईल? आता पुन्हा हा दुसरा कोडॉन तयार होईल CGC, हा दुसरा कोडॉन असेल. आता तिसरा कोडॉन कुठून तयार होईल? इथून G पासून GCG, हा तिसरा कोडॉन असेल. ठीक आहे, त्यानंतरचा कोडॉन कोणता येईल? इथून पुढे CGU. So it is the fourth codon. म्हणजे या पद्धतीने इथं जे काही कोडॉन्स तयार होतील, these codons, these are the, it is the overlapping condition. ही जी कंडिशन कशी आहे? It is overlapping condition. Overlapping. But genetic codon are non-overlapping. So this condition is non-overlapping. Okay, so here the genetic codon are always, that is non-overlapping. ते non-overlapping मध्ये असं सलग 3 सक्सेसिव्ह न्यूक्लियोटाइड मिळून एक कोडॉन तयार होईल, त्यानंतरचे पुढचे 3 मिळून एक कोडॉन तयार होईल. यातला कुठलाही कोडॉन पुढे येणार नाही किंवा कुठलाही नायट्रोजन बेस हा पुढे जॉईन होणार नाही. दॅट इज दे आर नॉट इन ओव्हरलॅपिंग कंडिशन, दॅट इज दे आर नॉन ओव्हरलॅपिंग. जेनेटिक कोड आर नॉन ओव्हरलॅपिंग. कन्सेप्ट लक्षात येतोय. सो इट इज द थर्ड कॅरेक्टरिस्टिक, that is the genetic code are non-overlapping. Then next characteristic, that is the genetic code are always commaless. So see student, that is it is the fourth characteristic, that is genetic code are always genetic code are always commaless. Genetic code are always commaless. So commaless means without any gap. That is, there is no gap between the successive codons. No gap between successive codons. No gap between successive codons. So see, that is here how they are. So here see, that is the 5 prime end and here is the 3 prime end. Okay, so here in the genetic code, that is they are, see here this codon being like this. So here A, C, G, U, G, C, C, G, U. So these are the genetic code. So here the 3 successive nitrogen bases, it is one codon. Then again the 3 successive nitrogen bases, it is another codon. So this codon specify one amino acid, this codon specify another amino acid. So here there are the 3 successive nitrogen bases. These are known as the codon, but in the codon there is no gap. There is no gap. So no gap between successive nucleotides. That is here the no gap. No gap between successive nucleotides. No gap between successive nucleotides. म्हणजे या न्यूक्लियोटाइड्स मध्ये गॅप नसतो, हे सलग न्यूक्लियोटाइड्स असतात. आणि लक्षात, सो फर्स्ट 3 that is one codon, then 3 another codon, then 3 another codon, like this. Okay, so the genetic codon, there is no any gap between the successive codons. So the genetic codes are commaless. Genetic code कसे आहेत? Commaless आहे. Okay. Then next characteristic, that is the genetic code has a degeneracy. Next, fifth characteristic, that is the genetic code has degeneracy. Genetic code has degeneracy. So here see, that is a degeneracy. Genetic code having the degeneracy, and here it can be explained with the help of Ubel hypothesis. It is explained by the Ubel hypothesis. So Ubel hypothesis means what? That is the third pairing of the codon and anticodon is unusual. That is the Ubel pairing असे म्हणतो त्याला आपण. So here genetic code has a degeneracy. हा concept जो आहे तो concept कशाशी related आहे? It is related to the Ubel hypothesis. So in Ubel hypothesis, that is, if we see here in total codons, that is in the chart you can see, that is the total number of codons, there are 64. There are 64 codons. And these 64 codons, these are mainly 64 codons. And how many amino acids are? 20 amino acids. So these 20 amino acids, these are coded by the 64 codons. Okay, so here one amino acid that is it can be coded, that is the 3 codons. म्हणजे generally एक amino acid, one amino acid can be coded, that is there is a possibility of the 3 codons. म्हणजे जवळजवळ कोडॉन्स किती आहेत 64 आणि अमायनो ऍसिड्स किती आहेत 20. म्हणजे जवळजवळ एक अमायनो ऍसिड हा किती कोडॉन्स कोड करू शकतो 3 कोडॉन्स कोड करू शकतो. ओके, सो हियर द वन अमायनो ऍसिड, फॉर वन अमायनो ऍसिड देयर आर द 3 कोडॉन्स and that is they are the possible. So here that is one amino acid can code the 3 codons. लक्षात येते का बघा, one amino acid can code 3 codons. One amino acid can code the 3 codons during the protein synthesis. So if we see, that is in the mRNA and the tRNA, so how the nitrogen bases is here. आता उबल पेरी किंवा उबल हायपोथेसिस हा कन्सेप्ट बघूयात आपण. So what is the Hubble pairing or the Hubble hypothesis? So see, that is here, it is the tRNA. So here the tRNA. So in the tRNA, on the tRNA there are the anticodons. tRNA वरती काय असतात anticodons. आणि mRNA वरती काय असतात codons. So here on the tRNA the anticodons, these are mainly read, that is from the 5 prime end to the 3 prime end. म्हणजे आपण जेव्हा tRNA वरती कोडॉन अँटीकोडॉन्स रीड करतो, these codon anticodons, these are read, that is from 3 prime end to the 5 prime end. And these are mainly, for example, on the tRNA here the anticodons, these are the A, G, U, these are the anticodons which are present on the tRNA. Okay, while the tRNA, when pair, that is this anticodons pairs with the codons, that is here the codons which are present on the mRNA. Okay, so it is the mRNA, and on the mRNA codons are present. These codons are always read, that is from 5 prime to the 3 prime end. Codons can be 5 prime end to the 3 prime end read. So here the codons, these are complementary to the anticodons. So if here is the adenine, here the adenine always pairs with the uracil. Okay, so here the uracil will be the here. Then guanine pairs with the cytosine. So on this mRNA there is the cytosine. While here is the uracil, it pairs with the adenine. So here the anticodons on the mRNA are the UCA. Okay, so here it is the usual pairing, that is adenine pairs with the uracil, guanine pairs with the cytosine, or uracil pairs with the adenine. So this pairing is a usual pairing. This pairing is usual pairing. So here next, that is here, according to the Hubble hypothesis, that is in between the codon and anticodon there is unusual pairing. म्हणजे जर Hubble hypothesis नुसार बघितलं तर इथं unusual pairing येईल. So see, that is here the tRNA, and this tRNA, if there are the anticodons, that is AGU, anticodon, that is the AGU, then what will be the codons in the the brassicus, the guanine, it is the unusual pairing. So here this unusual pairing or the oval pairing, that is it is possible. मग याच्याबरोबर अजून unusual pairing मध्ये इथे codons कोणकोणते असतील? That is codons in the mRNA. या mRNA मध्ये अजून codons कोणकोणते असतील? तर adenine आहे, guanine आहे, बरोबर आहे? आता आपण 4 न्यूक्लियोटाइड्स बघितले, adenine, guanine, cytosine आणि uracil. ठीक आहे? आता इथे adenine आहे, इथे guanine आहे. राहिलं कोणतं? cytosine. तर आता UCA, UCG झालं. ठीक आहे? राहिली कुठे आता? UC adenine झालं, guanine झालं, UCC. बरोबर? cytosine आणि UC काय राहिलं अजून? UC. Okay, so here these are the codons. These are the degenerating codons. These are the degenerating codons. म्हणजे ते codon जे आहेत ते are in degenerating state. Actual codon कोणता असेल? A, G, U ला या TRNA आहे आणि तो mRNA वरती usual pairing मध्ये कोणता असेल? UCA. ओके, पण जर आपण पाहिलं तर इथं पाहिलं तर इथं मात्र आपल्याला अनयुजुअल पेअरिंग दिसेल आणि याच्या बरोबरच या mRNA वरती अजून कोणती कोडॉन असू शकतील? दॅट इज UCG आणि UCU. ओके, UCG काय केलेलं आहे? UCC आणि UCU. लक्षात येत आहे? UCA घेतला, UCG झाला, UCC आणि UCU. So these are two codons. These are the mainly the possible codon to the TRNA. म्हणजे या TRNA साठी हे दोन आहेत ते possible codon. They are in the degenerate. They are not there. They are degenerating state. म्हणजे जनरली हे जे आहे ते इथे आपल्याला दिसत नाही परंतु ते कसे आहेत? degenerating state. ओके, so here that is genetic code has a degeneracy. म्हणजे युजुअल पेअरिंग सोडून इथं अनयुजुअल पेअरिंग मध्ये सुद्धा हा mRNA आणि TRNA हे पेअर होऊ शकतात. That is the third base pair is unusual. Okay, so here the genetic code has degeneracy. So we will see that is the fifth characteristic, that is the degeneracy. The next characteristic, that is the genetic code are universal. Genetic code are universal. Genetic code are universal. So universal means what? That is here the genetic code. These are having the initiation codon and termination codon. So see that is here the mRNA. So in mRNA there is the 5 prime end, there is the 3 prime end. So at the 5 prime end there are the initiation codon. Here are the initiation codon. So AUG, it is the initiation codon. Okay, while here this initiation codon, that is AUG, codes for amino acid methionine. So this codon which is present at the 5 prime end, it is known as the initiation codon, and this AUG codes for amino acid methionine. Amino acid कोणता amino acid कोड करता? methionine. So this methionine, that is it may be seen from the bacterial cell up to the human being. Bacterial cell up to the human being, that is in all organisms same codon, that is AUG, it codes for the amino acid methionine. म्हणजे कोणत्याही ऑर्गॅनिझम मध्ये पाहिलं आपण बॅक्टेरिया पासून ते ह्यूमन बीइंग मध्ये in all organism the initiation codon, that is AUG, only codes for the methionine amino acid. So here the genetic code, they are the universal. Okay, so they being universal. So in all organisms, that is the initiation codon AUG. These codes for amino acid, that is the only methionine. Okay, then so it is about the genetic code are the universal. Then next characteristic, that is the genetic code are non-ambiguous. Genetic code are non-ambiguous. Genetic code are non-ambiguous. So here, that is the genetic code not having more than one meaning. Non-ambiguous means what? Genetic code not having more than one meaning. Genetic code not having the more than one meaning. Specific codon codes for specific amino acid. Specific codon codes for specific amino acid. That is, we have seen that is the AUG, initiation codon. It is the specific codon, and this is the initiation codon. It always specifies only the amino acid, that is the methionine. Like this, that is here the specific codon codes for the specific amino acid. So here the genetic code, that is two different amino acid, does not code a single codon. Two different amino acid does not code single codon. Two different amino acid does not code सिंगल कोडॉन. म्हणजे एकच कोडॉन हे दोन डिफरंट अमायनो ऍसिड कोड करू शकत नाही. आलं लक्षात? एकच कोडॉन दोन डिफरंट अमायनो ऍसिड कोड करू शकत नाही. आलं लक्षात? सो हियर द जेनेटिक कोड इज नॉट हॅव्हिंग मोर दॅन वन मीनिंग. म्हणजे जेनेटिक कोडला एकापेक्षा जास्त मीनिंग नाही. याचा अर्थ काय? तर दोन डिफरंट अमायनो ऍसिड हा सिंगल कोडॉन कोड करू शकत नाही. Okay, so here the genetic codons, they are non-ambiguous. म्हणजे genetic codon हे non-ambiguous. म्हणजे काय आहे तर जनरली एकच कोडॉन हा दोन वेगवेगळे अमायनो ऍसिड हे कोड करू शकत नाही. ओके, परंतु एक अमायनो ऍसिड तीन कोडॉन मात्र रीड करू शकतो. आता जसं आपण ambiguous मध्ये बघितलं की कोडॉन्स आर डिजेनरसी मध्ये बघितलं तर आपण फक्त इथं काय होतं तर शेवटचा जो काही नायट्रोजन बेस पेअरिंग आहे तो बदलतो. मग त्यामुळे इथं एक अमायनो ऍसिड चार प्रकारचे कोडॉन कोड करू शकतो. परंतु एक कोडॉन मात्र वेगवेगळे अमायनो ऍसिड कोड करू शकत नाही. हे लक्षात ठेवा. Okay, so two different amino acid does not code single codon. So the codons are non-ambiguous. Codons हे कसे आहेत? Genetic codon are non-ambiguous. Then next characteristic, that is here the genetic code, they are mainly having the initiation codon and the termination codon. That is the genetic code 8 point. That is the genetic code having the initiation codon and termination codon. So see, that is if we see the mRNA, that is mRNA in which having at the 5 prime end. See here, there is the mRNA, here is the 5 prime end, and here is the 3 prime end. So at the 5 prime end there are the initiation codon, and this initiation codon which are present at the 5 prime end, they are the AUG or GUG. These are the initiation codon. or these are also known as the nonsense codon. So these are the termination codon, that is UAA or UAG or UGA. UAA, UAG or UGA. These are the three codons. These three codons, they are always present at the 3' end. याच्या व्यतिरिक्त हे तीनही कोडॉन मध्ये कुठेही दिसणार नाही. ओके, सो these are known as the termination codon. These three codons are known as the termination codon. These are the termination codons. Okay, so initiation codons and termination codons. So initiation codon, which are the initiation codon? A, U, G and G, U, G. While the termination codons are UAA, UAG and UGA. They are always present at the 3' end. Initiation codon are always present at the 5' end. Then next, that is the codons are universal. आता हा कन्सेप्ट आणि हा कन्सेप्ट थोडा वेगळा आहे. जेनेटिक कोड आर युनिव्हर्सल असं म्हणतो आपण की ज्याच्यामध्ये इनिशिएशन कोडॉन जो मिथिओनाइन आहे तो प्रत्येक ऑर्गॅनिझम मध्ये बॅक्टेरियापासून ह्यूमन बिईंग मध्ये सगळीकडे आपण काय बघितलं तर तो सेम असेल. आता इतर सांगितलेले की कोडॉन्स आर युनिव्हर्सल, that is all organisms having the specific codons codes for specific amino acid. म्हणजे इथं कोडॉन्स आर युनिव्हर्सल म्हणजे स्पेसिफिक कोडॉन हे स्पेसिफिक अमायनो ऍसिडच काय करतील कोड करतील. त्याच्या व्यतिरिक्त वेगळं कुठलं अमायनो ऍसिड ते कोडॉन स्पेसिफाय करू शकत नाही. Okay, so the genetic codons are universal. And third, that is there are the codons and the anticodons. That is here we have seen already, that is on the tRNA. If we see the tRNA, on the tRNA there are the anticodons. Anticodons. So see, that is here the tRNA. It is read 3' end to the 5' end. So on the tRNA, that is the nitrogen bases present, that is here they are the anticodons. And these anticodons, any anticodons, that is for example A, G, U, these are the anticodons. So the complementary, there are the codons which are present on the mRNA. Okay, and this mRNA, the codons which are present, these are the 5' end to the 3' end. And मग हे कोडॉन्स जे असतील, these are the complementary, that is the uracil, cytosine, and uracil is paired with the adenine. Okay, so here the codons and the anticodons, that is codons are read in the direction. Codons read कसे करतो आपण? Codons which are present on the mRNA. हे जे mRNA वरती जे असतात ते काय आहे? Codons. And on the tRNA, these are the anticodons. On the tRNA, these are the anticodons. On the mRNA, these are the codons. So here, the codons and anticodons, these are complementary. So anticodons are read in 5' end to the 3' end, while the codons, these are read in the 5' end to the 3' end. बघा, codons हे 3' end, sorry, anticodons 3' end to the 5' end present on the tRNA. हे कुठे present असतात? tRNA. Codons which are present on the mRNA, and they are always read from 5' end to the 3' end. Okay, so here the codons and anticodons, they are always complementary. म्हणजे codons आणि anticodon हे complementary असतात. म्हणजे इथे जर adenine असेल तर तो नेहमी काय असेल तर uracil असेल, guanine असेल तर cytosine, uracil असेल तर adenine. या पद्धतीने codon आणि anticodon हे कसे असतात? These are नेहमी complementary. Okay, so here the codons of mRNA and they are complementary to the anticodons on the tRNA. Okay, so it is about the 10th characteristic, that is the codons and the anticodons. Okay, so student, today we see that is about the characteristics of the genetic code, and there are the 10 genetic characteristics of the genetic code. That is codons are triplet, then codons having polarity, then codons are non-overlapping, codons are commaless, then codons having the degeneracy, they are universal, they are non-ambiguous, then there are having the initiation codon and termination codon. Codons are universal and codons are there are the anticodons. Codons are present on mRNA, anticodons are present on the tRNA. Okay, so today student will stop here.